the first slide. Cool. All right. So yeah, first of all, thanks to the organizers for inviting me here. And yeah, as I think you can see from my title slide being pretty blank and not containing any pretty pictures, this is an emergent project using an emergent method. So it's very much ongoing and under construction. So I welcome any feedback and comments and opinion that you have. It's very valuable to us in this stage, I think. Okay, so I think we've heard some about chromatin dynamics in this meeting so far. Yesterday there were some questions as to what do we even expect for what this looks like. So I have a plot for you. And here what we see is a mean squared displacement for histones in mouse embryonic stem cells. And why I'm excited about this plot is because by now we're able to track these guys over 6 orders of magnitude in time. So we really get a lot of sort of deep insight across different time scales into the motion of these histones. There's a sort of the cartoon on the right here is supposed to show that we put a fluorescent tag on histones and then we track them. In a nucleus or in a microscopy image it would look something like this. You get sort of a few fluorescent spots per nucleus. And then we can calculate a mean squared displacement, which I think was introduced somewhere in the previous days. Basically, the idea is to see over a given length of time, how far do loci move on average. And the first thing I want to do with this curve is put it on a linear scale by just taking a square root. You can see the curve is basically unchanged, just so that we can get an idea of what length scales really we're talking about here. So what people like to do with these MSD curves, and I also want to start out with this in a sort of hand-wavy argument way, let's put some lines on it. First one is up here. We see at sort of reasonably long scales, we see subdiffusive behavior, where if you believe this line that I put in here, that has an exponent of 0.4 in the MSD, it's a bit below what we see, what we expect for a Rouse model, but generally sort of roughly what we expect for a polymer. Loci on a polymer move subdiffusively, that's what we see here. Now at short times, we actually get diffusive motion. With this other orange line here. The diffusion constant I put here, just sort of as a note, all these curves are just overlaid by hand. This is not proper fitting. I will get back to why that is. The diffusion constant we get here is relatively low and noticeably much lower than what we would expect for example, free histones in a nucleus. The crossover between these lines is at 30 nanometers, which if you look in the cartoon over here, that has a sort of suggestive chromatin fiber. I think of that as quite exciting because the chromatin structure at these scales is very much unknown, and the idea is that here, by probing the dynamics of individual nucleosomes at very fine resolution, we might be able to make statements about the structure of the fiber. I will also get back to that later. The last line I want to put here is down here, just emphasizing that sort of in the MSD, usually what you see towards short time scales, you will run into the localization error of your method, which here is in the nanometer, seems to be in the nanometer regime. So this is kind of cool. Where does this data come from? The two curves up here, you can see span sort of somewhere around 2 orders of magnitude. This is conventional single particle tracking with different lag times in the movie. Here is a movie of how that looks like. This is sort of pretty much a standard technique. The people who were here on Monday for my workshop will know this movie. I talked about how we conventionally do the tracking in these movies. So that's all reasonably well known. This blue curve is from a technique called MinFlux, which was developed, was published in 2017, and you can see that it allows it gets us significantly higher time resolution. We're below millisecond time resolution here. This is sort of somewhere around 200 microseconds. The scope in principle can go a bit faster than that. And it also gives us 4 orders of magnitude of dynamic range, which to people familiar with single particle tracking is probably quite impressive. So So the first thing I want to do here is tell you more about this method, how it works, and give you an idea of what we're doing there. To do so, let's start out with how the conventional tracking works as a very brief overview. Here are snapshots from the movie that I showed before. You can identify by eye sort of one particle here. We can run a localization algorithm on this, and in each frame individually localize this particle. And then we sort of, in a post-processing step, if you want, we link these localizations together into a trajectory. And it looks something like this. And then we can run our analysis on these trajectories. As I pointed out, here we are independently localizing the particle in each frame of the movie, and due to how the physics of single particle fluorescence works out, the error you make in estimating the position of the fluorophore here is proportional to 1 over the square root of the emitted photons of the fluorophore, which means which is usually with acquisition settings reasonably optimized in these movies is somewhere in the thousands to 10,000 photons per spot per frame. We link them into the trajectories after the fact, and the main limiting factor in these methods, and why in the plot I showed you in the beginning we get about 2 orders of magnitude of dynamic range, is the photon budget of the fluorophore. So the fluorophores that are commonly used in these experiments have a limited, basically the way I think of it is they have a limited number of photons they can emit. After that they break, they basically change their fluorescent state, and you won't be able to see them anymore. So because you use somewhere around 1,000 to 10,000 photons for each of these localizations, you're limited in the length of the trajectories that you can get out of this. Okay, so that's the conventional picture. Now let's get into what MinFlux does. And for that, let's do a little thought experiment. Let's rewind time on this trajectory that I showed you. So we start with the fluorophore at the beginning, and I've outlined you here the trajectory that this particle will take. This is, of course, as I said, a thought experiment. The point I want to make is that if we pretend that we know, if we were to know exactly the trajectory that this particle takes, we can confirm it experimentally without investing any photons at all by using a donut-shaped excitation beam. So we use this light pattern of excitation that has a central minimum where the fluorophore is not excited. You can see I put it gray, it's dark now, we can't see it. And what we can do is we can basically just trace this a priori known trajectory of the fluorophore with the donut. And as long as we don't get any emission, we have a confirmation that we actually correctly predicted the trajectory. One fun side note on this is that now, because we're kind of quote tracking the fluorophore with the minimum of this beam, the localization error does actually not depend anymore on the number of photons that the fluorophore emits, because it doesn't emit any. Instead, it depends on the precision with which we can determine the central minimum of the beam, which depends on the number of photons that come from the laser, which is virtually unlimited. So that's kind of cool. But now, of course, in reality, this is not what's happening, because in reality we don't know how fluorophores move in advance. But we can exploit the basic idea in this thought experiment because we usually have a reasonable idea of where they are. And so the idea is to basically just incrementally update that knowledge. So here in this cartoon, I'm giving you the last position that we know of the fluorophore because we localized it before. And of course, since then, it's moved a little bit onwards in this trajectory. And now what we can do is we target this donut excitation pattern to the last known position of the fluorophore. And that will give us a few photons from the fluorophore, because now the fluorophore is somewhere a little bit off the minimum of this beam. And because we know the beam shape, we can estimate how far from the minimum the fluorophore should be. That puts the fluorophore position on this blue circle here. And essentially then you can just raster around this last known position with a few different positions of your excitation pattern. and you can triangulate the new position of the fluorophore. So here on the right, I show you the snapshots from this, in this cartoon I'm using 4 positions. The numbers down here are made up because it's a cartoon, but they are realistic. The statement is, to update the position of the fluorophore, we're using somewhere in the order of tens of photons instead of thousands or tens of thousands with conventional camera-based tracking. So the idea is that here we're utilizing we can triangulate the new position. And the idea is that we utilize the photon budget of the fluorophore more efficiently. Because we're only doing these incremental updates and not sort of localizing independently in each frame of a movie. Okay, so here's an overview over how this works. We just went through this. We sort of given a last known position, we raster around in some pattern. What we actually end up using in the experiments is a hexagonal pattern. After rastering around this and collecting photons from each of these positions, we can triangulate the new position of the fluorophore and then cycle through this rhythm. Of course, to do that, we need we need sort of to initialize the whole thing with somehow we have to know where to start. There's different ways of doing this. You can do a wide field image where you do sort of just conventional localization, or you can basically zoom this excitation pattern here. You see I'm giving the parameter, the size of this excitation pattern as a parameter. This is of course something you can change. If you start with a very big pattern, you get a very rough localization of your fluorophore, but you will get some signal, and then you can kind of zoom in incrementally to figure out where it actually is, and then start this tracking routine. There's one additional arrow I want to put on this plot, which is that if for some reason during this rastering step here you don't collect enough photons to reliably localize your fluorophore, you basically just do the same thing again until your fluorophore shows up again. This is very similar to conventional tracking, which I'm mentioning down here, where if you think about what this cycle here does, it's really very analogous to again, sort of for those who were here on Monday, I talked about this the conventional way we track these fluorophores. You use some linking algorithm. It typically has a cutoff in length, which we call a linking distance. And it has some memory where if the fluorophore disappears for a few frames, for example, because fluorophores blink, it might run out of focus in Z, or at some point it bleaches and then it's just gone. But as long as it just disappears for a few frames, we usually bridge these gaps. And so yeah, this is as I mentioned, this is basically very analogous to what we do in conventional tracking. The difference being that here the tracking really happens during the experiment. There's no camera involved in any of this acquisition. It's literally just photon counting. So you don't get a movie out of this. You get photon counts and particle localizations. Now, this method is still limited by the photon budget of the fluorophore, but because we're using way fewer photons, it's about 100 times more efficient, which means your trajectories get 100 times longer, which is quite cool. And overall, this has been shown again in the original publication, which, by the way, I'm like explaining this method here because I am excited about it, and I think it's a very exciting upcoming method. I had nothing to do with the original development. What they show is that it provides nanometer resolution in space with sub-millisecond lag time. So it really allows us to very fast and very precise track things in living cells. So what does this data look like? Here's a trajectory where we tracked a single fluorophore attached to an H2B on over about 4 seconds. And if we zoom into this a little bit, between the gray bars here, we can zoom in here and there. What we can see is these kind of gaps here. So this looks a little bit suspicious because it kind of looks like we're just blocking out a part of the trajectory where the particle kind of jumped away, and then we connected with a straight line. And you can see that this happens in various places in the trajectory. So here and there and there and there, we get relatively long gaps in these trajectories. And the kind of issue I had when I came across this is because this min-flux method is very close to sort of optimal information usage from each single photon you get, there's no redundancy. You don't have a movie that you can go back to and look at what actually happens there. The only thing you get is this trajectory. And after doing some more analyses, what we figured out is that likely what's happening here is that this linking radius that we chose is too small. And yeah, so sorry, let me, which means that sometimes the particle we're tracking jumps out of this linking radius, and then it comes back, we just connect the trajectory, but what that means is we're very heavily biasing against large steps of the fluorophore. Now, in some experiments, this is not actually a big issue, because if you were tracking, for example, a diffusive particle, or an even more extreme example, something that moves ballistically, if your linking method can't pick up large displacements, you basically cut your trajectory here, and your particle will never come back, it's just gone. So if you choose your linking radius too small, you will just get very short trajectories, and it's very noticeable that your parameter settings are not quite useful. Same is true with diffusive trajectories, but what we're tracking here is a histone, which is a locus on a polymer, moving somehow subdiffusively with a relatively small exponent. The MSD exponents are listed up here for these different trajectories. And again, sort of pushing this to the extreme would be a completely flat MSD, which just means your fluorophore appears around a fixed point in space. And you can imagine that this one, even if you throw out all the large displacements, you can perfectly track it for infinity because it never moves away, really. But you will miss all the large steps, and that will heavily bias your results. So the precise statement is that for an MSD with an exponent smaller than 2 over the spatial dimension, the motion that you expect from your particles is recurrent in time, meaning whenever you find it at some point, it will come back there eventually. But you might miss the excursions it does in between if you have too small a linking radius. And so what this means at the end of the day is that if we're tracking particles that exhibit this recurrent motion, so sort of small exponent in the MSD, and we use a linking algorithm that has a small cutoff and allows for large gaps, sort of has a long memory, then we can get long trajectories out of this that have a heavy selection bias in the steps that they take into account. So this is kind of 3 different conditions that have to come into place and happen to play out in the scenario that we have here. I can quickly show you that we can just check in simulations. If we run this min-flux localization algorithm on simulated trajectories, we see exactly this effect that the MSD is biased towards short distances, even though truly our particles move significantly faster than that. But we will still sort of be able to track it through the whole length of the trajectory. So what this shows is that, I don't know, I mean this orange plot here might be reminiscent of the plot I showed in the beginning, where it kind of transitions from one scaling regime to another with some sort of hump in between. Our current understanding is that that's exactly what's happening in our data. So here it is again. That's a bit unfortunate. We were very excited when we sort of saw this change in behavior. We thought maybe we could get some structural insights out of this. I still think that is true. We basically just have to fix this issue with the small linking distance. And what we do have in this data set is sort of a relatively sparse sweep of this parameter. Here's what it does. If we increase the linking distance, the MSD at short times here goes up, and we have some, very rough estimates that with a distance of around 200 nanometers, we might be able to track this without the selection bias issue. If you, I don't know, just very roughly by eye, it looks like we might be converging to something like this, which is just a straight power law over 6 orders of magnitude. That by itself, I think, would be very interesting. we've seen a lot of talks, so this is actually my next slide. Why do we care about any of this? Why do we care about tracking histones over different timescales? Here's a cartoon from an old review from 2007 that contains this 30 nanometer fiber that I think by now most people in the room are aware is not really what we believe in anymore. The question is, what do we put in its place? I alluded before that the chromatin structure at this sort of intermediate scale, sort of a few histones to say 10 kb or something, is something that we have really very little understanding of. On single nucleosomes we can do X-ray crystallography. On larger scales we can do high-C and oligopane techniques and so on. This intermediate scale is really very, very unknown. There are various works suggesting different organization. I want to point out the nucleosome clutches that I was hoping we could see in this change in behavior in the MSD. The point being, currently there's a lot of activity in the field trying to understand the structure at these scales with structural methods. And I think that, and I don't know, I hope I could illustrate that there is a fair hope that using this highly resolved dynamical method, we can take an orthogonal point of view on these unknown structures, which would be very exciting. The point being that if these clutches are a salient feature in the nucleus, we should see a dynamic signature of that showing up in the MSDs that we get. Yeah, with that, I think I just told you that the results I showed at the beginning were wrong. We made a technical error there. Stay tuned to get real results. We're basically going to redo the experiments and see what we get out of this if we do it properly. With that, let me thank the collaborators who made all of this possible. Anders Hansen at MIT, the driving force behind it, and his student and postdoc, Dominic and Matteo, who actually did the experiments together with Jessica Matthias at Iberia in Washington. Yeah, this is sort of directly collaborating with the company that manufactures the microscope. They gave us some imaging time on this, which we're very grateful for. And parts of Anders' work are also funded by NSF, which we're also very grateful for. Thank you.
Questions?
So it's very interesting, and I wonder to what extent increasing the linking distance will cause other problems by mistaking different spots that, like mistaking one spot for another one.
So, because you have this giant excitation, you have this giant donut beam, right, which is very large in size already. So because of that already, you have to do these experiments at very, very low labeling densities, like a few fluorophores per nucleus, which means you're not really running a risk of confusing fluorophores. You are increasing the localization error. So I think that once we calibrate this properly, we will not end up with the nanometer level resolution that I sort of showed on the cartoon in the beginning. We're probably getting closer to somewhere around 10 nanometers. But that's fine because the reason we're doing this is because the nucleosomes are moving that fast in the first place. So our localization error will still be lower than what the nucleosomes are actually doing.
Very, very cool methodology. I was curious, you kind of suggestively pointed to 30 nanometers as being the interesting point on that plot, but I would wonder why you didn't also just point to kind of 1 second as the interesting point on that plot, as that's the time scale that we think that motors are kind of making their steps at, and why you think you might be able to extract structural information when there's going to be such a strong signal, as we know from the works of many people now, of dynamics on this MSD.
Yeah, so that's another thing that we might be able to pick up. The reason I focus more on the sort of general chromatin fiber structure than on individual motor activity is because we're not, in these experiments, we're not actually targeting the label to anything specific. We're just broadly labeling histones. So the results we get will be averaged across the nucleus, which means that we might be able, if we sort of look in detail with a few fancy statistical methods, if we look at these trajectories, we might be able to maybe identify individual events where a molecular motor kind of passed by the nucleosome that we're tracking. But in the first pass of analysis, sort of the level that I'm showing here, we're really going to get a completely averaged over the nucleus picture, where I think we might still see some signatures of motor activity in how these curves change. But I think the more salient feature we're going to pick up is probably the fiber structure itself. But yeah, that's of course something to look into. Thanks a lot. Yes.
Yeah, very nice. I was wondering, do you have any prediction of what you expect if the clutches exist, or what would you expect?
Yeah, so I don't have a very well-founded prediction, I would say. My prediction for what we should see if clutches exist is actually relatively similar to this. I would expect to see sort of an increase in the slope of the MSD at short times. Basically, the logic being that you have these blobs of nucleosomes within which nucleosomes might be a bit freer to move than in the whole polymer. So the idea is on short time scales, nucleosomes move within these clutches. On long time scales, the clutches as a whole move as a polymeric locus. That picture is relatively consistent with the plot we see here. As I pointed out, that plot is wrong. So stay tuned.
Thank you.
Hello.
Yeah.
Can one use, because you are probing distances which are comparable to the size of the nucleosome. Can one measure the orientation of the fluorophore, for instance, by looking at polarization? Or?
I don't think so, because here so I don't really think so for two different reasons. One is that the 2 nanometers that I point out here are a little bit fake, because I showed you that the curve that we get here is mostly due to omitting larger steps from the trajectory, and what we will end up with at the end of the day is probably closer to 10 nanometers localization error, so more or less the size of a nucleosome. The second reason is that here we're really just counting photons from this fluorophore, so we don't really have any orientational information in the first place.
Yeah, but you might be able to put a filter and just sacrifice half of the photon to get another degree of freedom, which is a rotational one, or not.
I'm not sure that's technically feasible. So I'm not really in the depth of how the hardware of this microscope works. What I do know is that they had to very heavily optimize it to get to these fast frame rates, because you have to do the localization live. So I'm not sure that's going to be technically feasible, but I'll bring it up with the Iberia people.
Thank you.
Yes, here.
Yeah,
very nice. So I was wondering, since you are tracking the histones over very different time scales, do you see things that we don't see usually? For example, in our case, we see 3 populations of histones that we can classify depending on their diffusive parameters. Do you see new populations that we have?
So for now, okay, I haven't done the analysis in depth because we realized that we have this technical issue. I did look into it sort of, for a couple days. I didn't see anything that looks clearly like different populations of histones. We do, of course, see heterogeneity, which is something that I think we see in every tracking experiment. Some loci move faster or slower than others. But I think of it more as a continuous spectrum rather than different classes of motion.
And when you do the tracking, and you say because it's a redundant motion, then if you lose the tracking, you expect to see the same molecule at the same place a little bit later. How can you be really sure that it's the same molecule?
Sorry, so is your point that if we do the experiment at different time scales, the curves should match up?
No, but when you explained something about the mean flux, you said that because the alpha is 0.6, or you said then the same molecule. When you lose the molecule, then you have you wait until it comes back and you continue the tracking.
Right. So the point, I think the answer to that is very similar to the other question, that the labeling density in these experiments is very, very low. So basically, if you have one nucleosome that you're tracking, if you find anything sort of reasonably in the vicinity, it's going to be the same thing.
Okay, so you avoid the mistracking by being super, super low in density.
Yeah.
Okay, thank you.
All right, let's move on to our final presenter.